Hi. This talks a little bit about who we are and who I am. And what it doesn't say is that I'm basically a full-time volunteer. I'm very lucky to be able to do that. And I also live in a small town in Oregon, which means you get 15 minutes with your doctor. And your doctor sees a whole bunch of different types of cancer, and your doctor doesn't have a lot of time to keep up on all of this, stressing the need to keep things simple. I'll be talking about the consumer perspective on these tests and give some real-world examples and a look towards the future. And I also had very bizarre travel yesterday, so I'm going to be looking at my notes. Okay, this is Belle, and she was posing for a colorectal cancer calendar called the Colander. I know, I know, colander. But so she was diagnosed with advanced stage colorectal cancer several years ago when she was in her early 40s. And I don't remember who, but somebody talked about treatment resistance. She became resistant to all the treatments that were an option for her. And so she entered a clinical trial, and the trial was looking at immunotherapy drugs. David mentioned this. And it required that her tumor have a specific genetic profile to get into the trial. And so she had the mutation. She went into the trial. This is Andrew and his dad, Henry, and Andrew was diagnosed with high-risk ALL, leukemia, when he was 13. And he had a genetic test that looked at a chromosome, and his doctor, who was a rock star, said, okay, you are at very high risk of having your cancer come back after it goes away for the first time. So as a result of that, you need a bone marrow transplant. So Belle and Andrew both made incredibly important decisions about their treatment based on a test, right? And so this is what really concerns me, and I think everybody here has raised this issue. I borrowed the slide from an N.D. Anderson researcher, and it talks about what's needed to make good decisions, and I want to focus on the need for data, that good decisions are informed and supported by best evidence, and that poor decisions have an adequate objective evidence. And so we're patients, right? Everybody in this room is a patient at some point in time. You go to your doctor, and your doctor says, get a cholesterol test. Do you say, well, which test are you recommending? Do you say, who's processing the test? How was the test developed? How was it validated? You don't think about that. Your doctor says you need this test to get the test, right? And the point that comes up with all of this is that these tests are incredibly complicated, and they need to be validated, they need to be accurate, we need to know that what we think we know, we actually know. And I'm going to give a couple of examples that have kind of been touched on. Cetuximab is an EGFR inhibitor, and so there are these proteins that stick outside of cells, and they're called receptors, and so this drug comes and grabs hold of the receptors, and so it can't multiply and grow. And the scientists involved said, okay, well, this is an EGFR inhibitor, therefore we should only treat patients who have high levels of the proteins on the outside of the cell. And everybody said, that's great. So that's what they did. And it was initially approved in 2004 for patients with metastatic colorectal cancer with high levels of those proteins. But then, and so here's the thing that's important, is patients with low levels didn't have access to the drug because insurers wouldn't pay for it, right? So people were kept off the drug. Come to find out that EGFR was the wrong marker. It is actually a different gene, a mutation in KRAS gene. So that you have patients who were denied access to a drug who might have benefited from it because we assumed we knew we assumed we knew the science and we didn't validate it. So this is, I learned a lot from this. Then you have BRAF mutations, and cancers that have BRAF mutations are really hard to treat. They're very resistant to treatment. Then melanoma, they found a drug. Okay, I have to. But yes, thank you, vemurafenib. It's not approved for colorectal cancer yet, so I have no clue. So vemurafenib really helped most patients, or a majority of patients with melanoma, and it was like, oh my God, that's great. And so the company immediately started research on a bunch of different body parts to see if it would carry over to the other body parts, which was really cool. Unfortunately, in colorectal cancer, it didn't pan out. There's more going on. So a patient with BRAF mutant melanoma or BRAF mutant colorectal cancer will not benefit from this drug alone. Well, why does that matter? Because I know patients who, after seeing the melanoma research, went out and had the tumor typed and paid out of pocket for this very expensive drug because they assumed that what happened in one body part would carry over to a different. And again, the need to validate and validate. So this is Andrew and his wife Alice. She's a vet. They have goats, chickens, horses, dogs. He's a statistician. He has no long-term cognitive defects, obviously, from his treatment, which this is the success of precision medicine. Right patient, right treatment, right time. Unfortunately, Belle passed away in February. What more can you say about that? But, and David talked about this, these are the results from the conference a couple weeks ago for patients with Bell's kind of genetic profile. And if you look under the MMR deficient CRC, there are only 13 patients, but the disease objective response rate, that means the tumor shrank, 62%, that's huge. I've been doing this for 20 years. I've never seen a number like that in colorectal cancer. The disease control rate, 92%, never seen it. I mean, my jaw hit the floor, and I tend to be very skeptical about these things, and so future research is being looked into for this. This is my last slide. It's a Zen saying. I'm from Oregon, we do Zen. But it's, talk doesn't cook rice, and I really want the Precision Medicine Initiative to cook and not just talk. Thank you.